So, good afternoon, students.
Good afternoon, sir.
Good afternoon. So, how is everyone?
Sir, we are good. How are you?
Yes, I'm also good. Okay, so I can see only 14, 15, 14 students have joined, but I think we
Good
can start. We can start. So, students, till this point, we have talked about history of immunology, right? And you all know it all started with the development of vaccine against smallpox, right? So sometimes students get confused with smallpox, chickenpox, cowpox. Okay, so make it clear in your mind these three poxes. Okay, there is one more pox, monkeypox. Okay, but obviously the more confusing ones are the smallpox, cowpox, chickenpox, right? So I'm sure you know now that smallpox was a disease in humans, right? Cowpox is a disease in cows, right? And if that cowpox virus it infects humans, it does not cause a severe infection, it just cause mild infection, okay? And it provides protection against smallpox, right? So therefore we were using the lymph from the cows, okay, who were infected with cowpox, and we were using that as a vaccine against smallpox, okay. Okay, so we also talked about uh, antigens, right? Because when we are talking about the immune system, who is going to activate the immune system, right? Our body has a system, but it has a function and it has to be activated by someone, and that activation factor is the antigen, right? So we have seen the different properties of antigens, right? And in one of the practicals, we have talked about serum, the different components of blood, so serum, plasma, and the RBCs, platelets, right? And the buffy layer, right? The buffy layer, it consists of the white blood cells, right? I hope you remember all these points, right? So today we are going to talk about, again, we'll start with antigens, right? And then we will also talk about haptens, which is again an antigen, but it is called as incomplete antigen, right? So why it is called incomplete antigen, right? So we will see that. So today we are going to talk about three terms. One is antigen, one is epitope, and hapten. Okay, so students, before I start with these terms, first I'll ask you how much you remember, okay? And then we will I'll show you a small video of antigen and epitope, okay? And then we'll also talk about the properties of antigen, which you have already read, right? Okay, so students, when you are giving answer, just switch on your camera. Okay, so my first question is, what do you understand by the term antigen? So students, start raising your hands. What do you understand by the term antigen? Okay, so I can see two hands being raised. So Devanshi,
Sir, am I visible?
not yet.
Um, so now,
Yes, so Devanshi, what is an antigen?
sir, antigen is a foreign substance that is induced in a human body which induces an immune response in the body for the production of antibodies.
Okay, okay. My next question is, so students, Devanshi said a foreign antigen which elicits an immune response for the production of antibody, right? So my next question is an antigen always a foreign substance? Uh, okay, I can see one hand being raised. So yes, Manaswani?
Sir, so am I visible?
Yes, yes, yes.
Yeah. So, an antigen doesn't always have to be a foreign substance. It could even be, any kind of, let's say, a cancer cell that is completely from within the body, right?
Yes.
So, it doesn't always have to be foreign.
Yes. Manaswini, can you repeat? You can switch on your camera, switch off your camera, and can you just repeat the answer?
Yes, sir. So, what I had said was that an antigen doesn't always have to be foreign. So, an example of this could be a cancer cell or an autoimmune disease that's completely from within your body. There's no invading substance from outside.
Okay, fine. So basically, there are two types of antigens, exogenous antigen and an endogenous antigen, right? This is very important to understand because later on we will be talking about the pathways, right? So if an external antigen is coming, it uses different pathway, right? And if there is an internal protein, it is processed in a different pathway. Okay? Okay, so now next thing is we have already talked about antigens, right? Now my question is what are the different properties of an antigen, right? So let me start the Microsoft Whiteboard and then I'll write on it, okay? So let me just start it. Okay, students, so is it visible? Whiteboard?
Yes, sir.
Okay, so we will be talking about antigen. We have already started this. Okay, now I'll be talking about the properties, right? You know the different properties. So just students, tell me what are the properties of an antigen that influence the immunogenicity? So we have already read 5 different points. Yes, Shakti.
Am I audible, sir?
Yes, Shakti, you can tell me 2 points and then Yuvraj can tell me 2 more points, okay?
Yes, sir. So first one is foreignness.
Okay,
So greater is the distance, phylogenetic distance between the two species, and greater will be its immunogenicity. So
yes,
first one is that, and second one is I think I remember complexity. So
okay.
yes, sir. So complexity means the complexity of the protein organization. So more is the complexity, more is will be the immunogenicity.
So you are talking about complexity of what?
Protein organization, sir.
Yes, okay. Yes, Yuvraj.
So, in addition with what just Shakti said, so the from my side, the first one will be the molecular size. The greater the molecular size, greater will be the immunogenicity. And similarly, when we'll talk about the chemical composition or heterogeneity, it says that greater is the heterozygosity, greater will be the immunogenicity.
Okay. So we have till this point discussed four properties. One is foreignness. Okay. So this so the students have also explained. So foreignness, greater is the genetic distance, greater is the immunogenicity, right? Then protein complexity, molecular size, obviously bigger the molecular size, a better antigen it would be, right? And this is one of the factor which is important when we talk about haptens. Okay, so we will come to haptens later. Okay, next is heterogeneity. So one more point that we discussed. Anyone? So there were a total of 5 points. Okay, does anyone remember susceptibility to antigen processing and presentation?
Yes, sir.
Yes, sir.
So who can tell that? Just explain in one line if anyone remembers that. Okay, yes, Shakti, go ahead.
So, sir, it was about MSC 1 and 2 cells, I think, are the cells.
Okay.
So that was about how they present the antigen to the T cells. That
Okay.
depends.
Okay. So in this case, we'll just talk about MHC class 2 molecule. This concept will be more clear to you later. Okay. But basically, how an antigen is processed, right? So some antigens may not get broken down, right? And they may not be presented to the immune cells, right? So they are bad antigens because they will not elicit good immune response, right? But there are maximum antigens which are broken down, right? And they are presented well and they generate a very good immune response, right? Okay. So now let me show you. No, okay. Before that, another term is so. Does anyone know what is an epitope? So we have already talked about antigen, but what is an epitope? Anyone knows this? Anyone?
No, sir.
Okay. Okay. I'll just first explain you in simple words. I think one hand is yes, Sam.
I can't recall much, but it was a specific site on an antigen
Yes,
which is
yes,
expressed.
right, Sam. So suppose this is your antigen, suppose this is a bacteria, right? So this is your antigen. This whole is your antigen, right? And this antigen obviously is having different kind of proteins on its outer surface, right? And it can have different kinds of epitopes on its surface. Okay, so you can see on an antigen there can be different epitopes, right? Now what happens? Antibodies which are formed, they are formed against a specific epitope. Okay, just for example, an antibody is produced against this green epitope, right? This specific protein, right? So this antibody will go and bind here. Now this antibody will only bind to this specific epitope. Okay. This antibody will not bind to any other epitope, right? Okay. So let's understand this concept with the help of a video. Okay. And then we can have more discussion on this point. Okay. Okay, so this video, it talks about first antigen, right? And then it also talks about epitopes. Okay, so let's see this.
In this video lecture we will talk about two terms which are often confused in immunology. These are antigen and epitope. Antigen is a substance which is identified or recognized by the host's body, or in our case, human body, as foreign, and, as a result, an immune response is stimulated towards it. Antigens can be foreign or self. Let us see what does it implies. Foreign antigens. Foreign antigens are also known as heteroantigens. As the name indicates, these substances are not part of human body. They originate from outside the body. They can be microbial agents. We are saying agents because these include whole microbes or their parts. For example, capsules, cell walls, flagella, fimbriae, toxins of bacteria, the coats of viruses, or any surface component of microbes. Foreign antigens can also be non-microbial agents. Non-microbial agents may arise from animals, plants, other humans. For example, pollen, egg white, serum proteins and blood cells from another individual, transplanted tissues and organs.
Okay, students.
Self-antigens. Self-antigens are also known as auto.
So is this clear? So when we talk about antigens, it could be the microbial antigens, okay? And when we talk about microbes, it could be any, suppose if not the whole bacteria or virus is entering, it could be even some part of that organism, okay? Because we actually just need a specific protein. So whatever protein this bacteria or virus have, it is foreign to the host, right? So even if you put a part of this into our system, so our system will recognize it as foreign, okay? So these are the microbial agents, and then you have the non-microbial agents, right? So there can be different, again, there are a lot of examples. One of the example is the pollens, right, which comes from the flowers, right? Uh, like product of other animals, right? And in fact, I don't know if you have heard about the pigeon groupings, right? So when the pigeon groupings they dry, right, they have some proteins in that. So if you inhale it, that can create lung issues, right? And another example that they have also talked about is any protein from any other person, right? For example, the blood, right? We have to first match the blood groups. We have to match the antigens, right? If the antigens are same, we can use that blood. If the antigens or the blood cells, if they are different, right, then uh, we cannot uh, transfuse that blood, right?
For example, pollen, egg white, serum proteins and blood cells from another individual, transplanted tissues and organs. Self-antigens. Self-antigens are also known as autoantigens. Although, as stated earlier, our immune system is able to distinguish between self and non-self, but sometimes our body provokes an immune response against its own cells and molecules. Rheumatoid arthritis is an autoimmune disease, in which immune system attacks and damages own tissues. Now that we understand the term antigen, let's move on to the next term, which is epitope. If you remember previous lectures, we said that the immune system does not recognize whole pathogen but portions or parts of these agents. We termed these portions or parts as antigens. Now, if we explore what is the chemical nature of these antigens, we'll find that most of these are large protein molecules, some are polysaccharides and a few are glycoproteins or nucleoproteins. Our immune system recognizes these antigens by recognizing three-dimensional shapes of regions called epitopes. They are also known as antigenic determinants because they are the actual part of an antigen that determines an immune response.
Students, so is this point clear? And it is also called as antigenic determinant because this is the point, or this is the point where your antibodies will come and bind. Okay, so they will not bind to the whole of the antigen, they will bind to the specific epitope. Okay,
Each antigen can have several epitopes. Let's understand this by taking an example. We know that most antigens are proteins. So when antibodies bind to the antigens, which are protein in chemical composition, antibodies do not bind to the whole protein, but they bind to a segment of that protein which is known as epitope. Now this segment consists of few amino acids. These few amino acids forms the epitopes to which antibodies bind. It means a single protein antigen can actually contain many different epitopes to which antibodies can bind. Epitopes can be defined as regions or sites of an antigen that are recognized by the immune system, and as a result, they bind to specific antibodies or T-cell receptors. So, we now understand that an epitope is a part of an antigen.
okay students, so is this clear? So now you understand what is epitope, what is antigen. Any questions at this point?
Sir, so does the epitope what gives the antigen its immunogenicity?
Yes, because see, as I said, these antibodies, they go and bind to specific epitopes, right?
Yes, sir.
Yes. Okay, any other question?
No, sir.
So I think now we can have attendance, and after that we can start with haptens. Okay, so Yuvraj,
Present, sir.
Yuvraj, you are not visible.
So my camera is on.
Yes, Yukta,
Present, sir.
so Yukta, again you are not visible.
Sir, my camera is on. Am I visible now?
No, you are not visible. Vivek, yes, Yukta, you are visible. Vivek,
Present, sir.
okay, Tridev, Tridev, okay, Tanvi,
Present, sir.
Tanvi, you are not visible.
Sir, my camera is on.
Shivani,
Present, sir.
Shakti,
Present, sir.
Sanjana,
Present, sir.
Sahiba,
Present, sir.
okay, Rupal, Nehal,
Present, sir.
okay, Nandini,
Present, sir.
okay, Namrita,
Present, sir.
Manaswini,
Present, sir.
Kshitij,
Present, sir.
okay, Kshitij,
Yes, sir.
Kshitij, you are not visible. Okay, Janvi,
Present, sir.
Ishita, Devanshi,
Present, sir.
हो गई बात, Sam,
Present, sir.
Ayushi,
Present, sir.
Atoiba,
Present, sir.
Adish,
Present, sir.
okay, and Tridev,
Present, sir.
okay. Okay, so students, now we are going to talk about another term which is hapten. Okay, now this is again very interesting. Let me share the slides. Okay, so when we talk about haptens, we also call them as incomplete antigens, right? So let's see why they are incomplete antigens. Okay, so haptens are antigenic, but they lack immunogenicity. So I hope you can understand this. There are two terms. One is antigenicity, another is immunogenicity, right? So in the very first class, I said that understand these terms, right? Because these terms will come again and again. So haptens are antigenic, right? But they lack immunogenicity. So they are not able to generate an immune response. Okay. So they can combine specifically with antibodies or surface receptor of T cells, but they are unable to induce an immune response on their own. Okay. So haptens basically they escape the immune system, okay, and therefore uh, they cannot induce an immune response on their own, okay. So that's why they are called as incomplete antigen. Now, hapten becomes immunogenic on combining a carrier molecule. Okay, so see now you can make, so hapten is not immunogenic, right? But we can make it immunogenic by combining it with a carrier molecule. Okay, so basically, uh, if you take, okay, let me explain it. Okay, so suppose you take a mice, okay, and you put hapten in this, okay. So in this case, antibodies will not generate, okay, because see hapten is not able to induce an immune response, therefore there will be no antibody against this particular hapten, right? But if you really want to have antibodies against the hapten, then what you need to do, you take this conjugate of hapten plus a carrier protein, okay. So now just take the mice, okay, and inject this into the mice, and then you will see a lot of different type of antibodies, okay. So now you can see different types of antibodies. I'll talk about how many different types of antibodies you'll find, okay. So is this clear? So haptens by themselves, they cannot elicit an immune response. But if you add a carrier protein to the hapten, right? So this becomes a hapten carrier conjugate, And when you inject this, then there is 3 type of antigenic determinants, right? So this haptane carrier conjugate will have 3 types of antigenic determinants or the epitopes. One is the haptane determinants, right? वो जो तुम्हारा haptane था, this one, okay? Unaltered epitopes on the carrier proteins, right? तो इस carrier proteins के जो epitopes थे वो, okay, and some new epitopes formed by the combination, right? So हो सकता है when we make this conjugate, some new type of epitopes are also formed, okay? So now 3 type of antibodies are formed accordingly. So now you can find antibodies against hapten, okay, antibody against the carrier protein, right, and antibodies against the conjugate of hapten and carrier protein, right? Clear? So basically, just for example, hapten का molecular weight बहुत कम था, right? So it was not visible to the immune system, right? But when you conjugate it with the carrier protein, right? So now the size has become bigger, it is visible to the immune system, and now immune system will make antibodies against the epitopes, and in that case it will also generate epitope antibodies against the hapten. Okay, now there is one example you can see. One of the example is DNP. Okay, I don't actually remember. It's dinitro something. Let me see. Yeah, this is dinitrophenol. Okay, so if you just add DNP, if you just inject DNP in a mice, you will not find any antibodies against DNP. Okay. Now, if you just add this carrier protein, for example, BSA, bovine serum albumin, you will find antibodies against BSA, right? Because obviously carrier protein is a big protein. It has the properties of good antigen. It will lead to generation of antibodies, but hapten by itself will not lead to generation of antibodies. Now what you are doing is you are uh, conjugating hapten and the protein carrier, right? Now after conjugating this, right, so there is this DNP BSA conjugate. Now you can see that there is antibodies against DNP, against BSA, and also if some other epitopes are formed, you'll find some antibodies against them also, okay, right? So by itself, hapten was not able to generate immune response, right? But when it was combined with the carrier protein, now an immune response is generated against this hapten, okay? Now haptens are used in research and diagnosis. It's used as a tool to study specificity of antigen antibody reactions, to demonstrate the diversity of antigens recognized by the immune system. Hapten specific antibodies are used to measure certain substances and hence can be used in diagnostics. Okay, so like in diagnostic, so if you are looking for a specific uh, antigen which is there in our body, right, but it is not visible because we don't have antibodies, so we can use hapten specific antibodies, right? So that can help in the diagnostics as well. Okay, so Landsteiner, he worked with these haptens. Okay, and I will also show you the picture of this. So what he did, he immunized rabbits with a hapten carrier conjugate. Okay, and he tested the reactivity of rabbits' immune sera with that hapten and other closely related haptens. and he observed cross reaction with haptens differing slightly in chemical structure, and by observing what modifications of the hapten led to cross reaction, he was able to deduce about specificity of antigen antibody reactions. So just by using haptens, he was able to study the interaction of antigen antibodies. Okay, so okay, I'm leaving this. Okay, now let me come to some of the examples of haptens. One we have already discussed, the DNP, right? So there are some other examples like fluoresceins, biotin, and penicillin. Okay, so now penicillin again, it's a very important example because see, penicillin is, what is penicillin? It's an, students,
Antibiotic.
antibiotic, right? So if you eat antibiotic, do you expect that our body should generate an immune response against it? Students, so students, my question is, if you are having antibiotics, right, does your immune system need to make antibodies against it?
No, sir.
No, sir.
No? So Manaswini, you said no?
Yes, sir. I mean, I said no.
Okay. Yes. See, because if you are taking antibiotic and an immune response is generated against it, what will happen? The immune system will take out that from the system. Okay. So we do not want that, right? Now coming to this. So by itself, penicillin is not antigenic. So when penicillin is injected in our body, it will not lead to generation of antibodies, right? Because अगर penicillin के against antibodies बनी, तो क्या होगा? वो antibody आएगी, penicillin के साथ bind करेगी और उसको system में से हटा देगी, right? और फिर penicillin का तो जो role है वो हो ही नहीं पाएगा, right? So it should be non-antigenic, right? But sometime when it combines with certain serum proteins of sensitive individuals, the resulting molecule does initiate a severe and sometimes fatal allergic immune reactions. Okay, so what happens in this case? In these instances, the hapten is acting as an antigenic determinant on the carrier molecule, right? This point is clear. So if you're injecting antibiotic, if you're injecting penicillin, right, it should do its work of killing the bacteria, right? And it should not lead to generation of antibodies, right? But sometimes what happens in some individuals, okay, so we will talk about it, why some individuals, right? Why not everyone, okay? But some people who are sensitive to penicillin, right? In them, what happens, this penicillin molecules, it binds with some other serum protein, okay? So now penicillin by itself does not generate immune response, right? But when it binds with some other protein, so this becomes a hapten carrier conjugate, right? And now the size has increased, right? And this is now visible to the immune system. Okay, so now antibodies will be formed against this carrier protein and against this penicillin, right? And this can lead to allergic reaction. Okay, it's not the allergic reaction that we see, right? So it's not the type 1 allergic reaction. So I'm just talking about it. I'll make it more clear when we talk about hypersensitivity, right? So sometime in these cases, hapten is acting as an antigenic determinant on the carrier molecule. So is this thing clear? Any questions on this?
No, sir.
Sir, I didn't get the allergenic reaction thing.
You didn't get?
The allergic reaction thing, how it's related.
Okay. Okay, so it's very simple. जब हम पेनिसिलिन का इंजेक्शन लेते हैं, right? That penicillin should not generate antibodies, right? Because अगर penicillin के against antibodies develop हो गई, then it will be taken out by the system, right? Antibodies का काम ही यही होता है कि उस प्रोटीन को सिस्टम से हटा दो, right? But in some people, right, who are sensitive, okay, so in those individual, what happens? This penicillin binds to some serum protein, right? Now, because of this binding, there is formation of a haptane carrier conjugate, right? So penicillin plus carrier protein, right? तो अभी जो हमने पीछे समझा था कि in this case जब तक पेनिसिलिन अकेला था देयर वाज नो एंटीबॉडीज, राइट? बट जब वो कैरियर प्रोटीन के साथ कॉन्जुगेट हो गया, नाउ एंटीबॉडीज आर आल्सो फॉर्म्ड अगेंस्ट पेनिसिलिन, राइट? एंड if an immune response is generated, then that can lead to allergic or hypersensitivity reaction in that individual, and which can even be fatal. Okay, so अभी इतना ही समझो, इससे ज़्यादा hypersensitivity की डिटेल्स हम hypersensitivity की क्लास में समझेंगे. ओके, okay, is it clear till this point?
Yes, sir. But how does an allergic reaction will be fatal? Sir? This part I didn't get.
ओके, वही मैं बता रहा हूँ कि ये हम जब हाइपर सेंसिटिविटी को डिस्कस करेंगे देन आई विल एक्सप्लेन, राइट? बिकॉज नहीं तो अभी मुझे तुम्हें टाइप 1, टाइप 2, टाइप 3, टाइप 4 सारा कुछ बताना पड़ेगा. ओके,
ओके.
उसमें व्हेन वी विल टॉक अबाउट दिस, सो वी विल टॉक अबाउट हाउ ड्रग्स कैन इंड्यूस हीमोलाइसिस. ओके, सो वी विल टॉक अबाउट दैट लेटर.
ओके,
राइट? और अगर फिर भी समझना चाहते हो एक सिंपल टर्म में, सो व्हाट हैपेंस, some of the components of penicillin, they will bind to the RBC, okay, and now an immune response will be generated against the RBC, and that will lead to lysis of the blood. Okay, and obviously अगर आपकी बॉडी के अंदर ब्लड लाइसिस हो गया, then it can be fatal. Okay,
yes sir.
okay, so just to make you understand this again. So when hapten is injected in a rabbit, you do not find any antibodies against the hapten, right? So no antibodies here, right? But if, and okay, now another case, you are putting this carrier protein, right? So when you inject this, you find antibodies against this carrier protein, right? But not against hapten. But now in this case, this is a hapten carrier conjugate. So on this protein, now you have the hapten attached, right? Now if you inject this, you will find antibodies against both uh, this carrier protein as well as hapten. Okay, so I think I'll just stop here, and after 5 minutes we have another class and we'll continue this discussion. Okay,
Yes sir.
so any question?
No sir.
So your students just think if there is any point you want to discuss again. Okay, students, so we again have a class after 5-7 minutes. Okay, so I will start with you. Okay, so you have to first explain me what is hapten. Okay, so we'll have a discussion. So now you all have understood what is hapten. So you make me understand what is hapten, and then I will show you a video on haptens. Okay, and once everything is clear about haptens, we will move to the next topic, which is antibody. Okay. So students, take a break. See